All right, so this is the lab that we are now working on, which is your separation of proteins by gel electrophoresis. You guys should be able to pull this up on Google Classroom. It's a fewer molecules that you guys are going to be putting into the gels this time. So unlike mysterious moving molecules where you guys filled all 8 wells, we're only going to fill 6 here. 4 with proteins that we know of. 1 will be a mystery protein. In the 6th well, we are going to put just a plain dye. It's kind of our control to make sure things are running. And what you guys are looking at within this particular lab, we're still running it based on charge. So whether we have positive or negative molecules, as well as molecular mass. The farther they move away from the origin, the lighter those particular molecules are. But with this particular lab, we are looking at something called the isoelectric point of a protein. So this isoelectric point of a protein is the pH at which the net charge of that molecule is basically neutral or 0. So pH at which the protein does not migrate to the positive or negative electrode. So of our 4 proteins that we're filling in, cytochrome C, myoglobin, hemoglobin, and serum albumin, you guys will see a little isoelectric point on that chart there. It's also going to give you the net charge of a pH about 8.6, which is about what our buffer is. So it's going to say whether it's negative or positive or very negative, and it should kind of give you guys an idea of which way things are going to be moving. So you should be able to kind of create a little hypothesis a little bit in this particular chart, meaning which way do you think the proteins are going to go? They're going to go to the anode, they're going to go to the cathode. Why? The only one that might be a little bit of a problem for you is mystery protein. It's a complete hypothesis. Which way? You don't really know what that mystery protein is. I'll give you a hint. It's going to be a combination of some of your other proteins kind of mixed together. It could be 1, 2, 3, maybe it's all 4 of them. And by breaking up those bands, you guys will kind of be able to see that. So with this, let's talk a little bit about some of these particular proteins that you're going to be looking at. So cytochrome C. Cytochrome C is found within plants and animals. It's part of the electron transport chain, which is the 3rd step of cell respiration. It occurs within the mitochondria, but this particular protein is going to help to produce or make that ATP within the mitochondria. It's pretty basic in nature, as you guys can see by the charts. Isoelectric point's about 10.2, so it's usually going to carry a positive charge. The next one that you guys are going to be filling in is known as myoglobin. Now myoglobin and hemoglobin, which is the 3rd one that you see, they're both part of the heme groups, which is kind of like if you think about the pigment within your blood or the iron carrying component within your body. This myoglobin, this heme group, is the iron involved in oxygen binding. It's going to store and transport oxygen in the muscle. A little different than your hemoglobin, which does it within the blood. This is going to store and transport oxygen within your muscles. So isoelectric point is about 7.2, and when we look at that one, it's also going to be negative. So hopefully we're moving towards that positive end of the box. Hemoglobin, we just mentioned, is also part of that heme group. This is a protein in your red blood cells. So if you think about like your blood, it gets transported to the heart and the lungs and back and forth, but it is transporting oxygen to the tissues within your body as the blood makes its way to those particular tissues. Its isoelectric point is going to be lower than myoglobin. It's about 6.8, and hopefully should move towards that positive electrode as well. Serum albumin. Serum albumin is a little different than your other 3 that we just talked about because this one is going to be completely colorless. However, a blue dye was added to it. So when you see the band, it's actually the blue dye that you have, but that blue dye is picked up by that serum albumin protein as we separate it out. This is a protein that's found within your blood plasma. It also has a very low isoelectric point and very negative net charge. That leaves you guys with your mystery protein. Like I said, mystery protein is going to be kind of a combination of a few of them here. And then, of course, we have that just plain old dye, which is our control that we're going to be looking at. So I'm going to load this up for you guys real quick. We're going to skip that first well. We're going to skip that last well. And that's going to put us our 6 within the middle. We're going to load about 8 microliters of each of them. So once again, my first one, let's see if I can find you guys here. My first one that you guys see is my cytochrome C. So I'm going to take my 8 microliters of my cytochrome C, and I'm going to skip that first well once again, and try to sneak over top of this so you guys are probably looking at the back of my head right now, and I'm going to slowly load that into there. Remember, we're using a fresh tip for each time, and you can slightly see that in there. I'm going to slide it off for you guys in a moment. once I get them all loaded. My next one, we are now looking at my myoglobin there. So myoglobin and hemoglobin, part of those hemi groups that transport oxygen throughout our body. This one is within the muscles. We're just going to go into the third well, because remember we skipped that first one. Cytochrome C is in the second well. You get a first push, get a second push, slowly put it in there. You guys can see that they're starting to fill in. My third well, we are now going to put in my hemoglobin. So here's my hemoglobin that you guys see here, kind of red in color. So you guys are going to kind of do what you did with Mysterious Moving Molecules. You're going to be filling in your data chart. Here's the kicker though. You're making your own data chart. I did not make you guys one for this particular one. Now that you know the information that you guys should be putting in there, you guys should be able to design your own. Maybe you like designing it better because it fits your way that you're able to see your information. So as we have separation, maybe make separate columns for those particular bands that you're separating out. Some of you guys like making your own charts. Maybe you see it a little bit differently than I do, and that's completely fine. Next, we've got serum albumin. So serum albumin is that colorless one. It's got the dye added to it, so it's got a blue dye to it. And that way we're able to see it move. Put this one right below my last one. This should be well number, or that's my fifth one that we're putting in, or my fifth well, I should say. Next, I'm gonna have my mystery. Mystery protein is a combination of a couple. 1, 2, 3. You guys are gonna let me know. You guys are gonna match up the bands. There's no name on it because it's just a plain old mystery protein. This is gonna go into my sixth well. Remember we left the first one blank. And then I'm just going to add plain old loading dye to the last one. It's kind of my control. I just want to make sure that it does work and that it is running. If that's working correctly, I know that my proteins are also working correctly. So that'll go my seventh well. Remember, my first well and my last well are my blank wells, unless of course you mess up and you need to utilize one of them. It goes back. So now that's what we have. I'm going to slide it off. There's my initial colors. Once again, first well, last well are empty. It's hard to see on that red line, so I'm going to kind of slide it over a little bit more for you guys. And there are my initial colors for you. Let's see if I can get you a little bit better view. There's my initial colors. The top is well 1. The bottom is well 8. And there are my colors that we have there. So now what's going to happen is we're going to kind of go through and we're going to hook it up to our electrodes just like we did last time. So the red goes to red, black goes to black. They get pushed in to the sides. I'm going to turn my gel box on. You guys can kind of see that off to the side. I got to set about 96, and that's just because we want to speed it up a little bit with the shortened class periods that we have. I'm going to shut it off real quick because what I need to do is I slid it off to the side for you guys to see the colors. I want to put it back on that little platform there just to make sure it is secure, and I'll plug it right back in again. So you guys are going to go through and do an initial sketch. You guys are going to do a 5-minute sketch. We're going to make sure things are running. At the end, about 20 minutes, we are then going to go through and say, okay, well, what does it look like? I can actually physically pull it off of the platform, take my particular gel out, and this is one that I already have done, and I can slide it kind of right in place for you guys. I already have my piece of paper, so there's my black end, my red end, well 1, well 8, as you guys can see there. And what they're going to do, and you're going to see the last picture like this, is that we're going to take our rulers, always in centimeters, and we're going to hook them up to the origin. So there's my first one, right there. And if I go the other way, go like right there. Put some boxes underneath, hold my rulers up. All right, so that's how you guys are gonna see that last picture. Remember, your solvent front is from the wells to the end of the gel. And yes, you need to measure in both directions because this one is a little bit longer than the other way. So going towards the anode is longer than the cathode. May not be true on the one that you have. Depends upon how I put that comb in there. It's got a little lip end, so if I turned it this way or did I turn it that way? Maybe this end towards the cathode is longer. You guys are measuring each band. So cytochrome C is moving towards my cathode. Measure it there. I have no separation. Notice my second and third ones, my myoglobin and hemoglobin. They are huge molecules, molecular mass-wise. They are not moving, so they're basically almost stuck within there. You see a little bit of movement on both, but they're barely coming out. My next one is going towards the anode. My mystery protein, you guys are going to see two bands. Yes, there's separation. Yes, I'll have two measurements. Yes, I'll have two RF values. Yes, I'll have two charges. One's going to the anion, one's going to the, or one is going to be an anion, one's going to be a cation, one's going to the cathode, one's going to the anode. I do not need you to measure out my dye. This is my control. So that is just making sure that everything is running. Yes, it's running. Yes, it's moving. So I don't need you to make me a column for loading dye. Loading dye is not a protein. So you guys are looking at cytochrome C. You guys are looking at your myoglobin, hemoglobin, serum albumin, and then you have within that fifth one that we put in there, that mystery protein. And you guys are going to kind of look at that and tell me what that mystery protein is. So if you're looking at this and going, oh my God, that's so hard to go through and measure for, I put on there a document that shows picture-wise, so you guys aren't going through and trying to measure off the video. What does it look like at the initial? What does it look like at 5 minutes? What does it look like at the end, roughly 20 minutes? Can you just copy and paste and put them into your particular lab? Absolutely. If you want to go through and sketch your own, do so. Okay, and then there'll be some questions that go along with that. Think about that mystery protein and combination of it. Make sure your data chart has all the information that you should have. Go back to Mysterious Moving Molecules if you have to. Let me know what questions you may have.